This is Rochelle, your Microbiology IT tutor. Welcome to today's Microbiology Retention session where we will be studying genetic information flow. If you need study resources to help you get that A, then grab your Microbiology IT workbook. The link is in the description box. So the first one is genetic info flow. There are 3 branches stemming from here. The first one is replication. And then from replication, we have 3 points stemming out. We have enzyme, and the enzyme involved is DNA polymerase. And we also have template, and we have the parental DNA strand that is the template used. So it's the parental strand. And what is the product at the end? Well, the product is the complementary daughter strand. So we're going to put up here daughter strand. All right, our next branch is transcription. DNA to RNA. We have enzyme, where the RNA polymerase is involved. The template is the DNA. What is the result? The product, messenger RNA, tRNA, transfer RNA. Let's transfer RNA and rRNA, ribosomal RNA. In prokaryotes, this occurs in the cytoplasm simultaneously with translation. So this happens in the cytoplasm. And then in eukaryotes, it happens in the nucleus right before translation. Okay. And then our last branch here is translation. 3 branches coming out of that one. And then we have, where is it located? Where is this process located? So we have location, and the answer is ribosome. What are the major players in this process? Messenger RNA, tRNA, and rRNA. Now, messenger RNA holds the codons, and there are a set of 3 nucleotides, and the tRNA holds the anti-codons, where they connect with codons. And then rRNA, its part is for peptide bond formation. And then our third branch here is a product. What happens at the end? Well, one, you will receive well, let's start with this one. You get a chain of polypeptides. So you have a polypeptide chain, which would result in a functional protein. So that is our micro mind map. Let's go over to our notes. So the enzyme, like I said, is DNA polymerase. It is used to help catalyze the addition of nucleotides to a growing DNA strand. We have a template, which is the parental DNA strand. The parental strand serves as a guide. We have the product, which is the complementary DNA daughter strand. This is the outcome. The outcome is a new strand that is complementary to the parent strand, forming a double-stranded DNA molecule. We went over transcription, the enzyme in play, RNA polymerase. The template is the DNA. The products are the messenger RNA, transfer RNA, and rRNA. And where is it located? Where is this process located? In the prokaryotes? Well, in cytoplasm, and in eukaryotes, in the nucleus. We went over translation, and that's RNA to protein. So we have a location, it's the ribosome. The translation takes place on the ribosome, where messenger RNA is encoded into a polypeptide chain. The players involved are messenger RNA, transfer RNA, and rRNA. The messenger RNA provides codons, transfer RNA provides amino acids through the anti-codon pairing with the codon, and the rRNA catalyzes peptide bond formation. What are the products? Well, you end up getting a polypeptide that transfers into a functional protein. So the final product is a polypeptide chain that folds and undergoes modifications to become a functional protein. So let's test your knowledge. Yeah, quick recall fill-in. Number one, the enzyme that catalyzes transcription is blank. And feel free to put your answers in the chat. And use the micro mind map right here that you've created. Number two, in prokaryotes, transcription and translation occur. Where does it occur? You want to go back to your notes and see where it occurs. Number three, the codon on messenger RNA pairing with the blank on tRNA. So the codon on the messenger RNA pairs with what on the transfer RNA? And that answer is in your notes on the micro mind map. So feel free, put your answers in the chat. We're going to move on to our next slide. We have the multiple choice questions. So we have number one, which of the following correctly describes the central dogma? A, B, C, or D? Number two, in eukaryotes, transcription occurs in the number three, which RNA type carries amino acids to the ribosome? So if you're getting stumped, go back to your micro mind map and you'll find your answers. And then you can place them in the chat. Fill the flow and challenge. So fill in the missing steps in the correct order. So we have DNA, then what is the next step? And then the next step. And then the last one is protein. Application. So a mutation occurs in a bacterial promoter region that prevents RNA polymerase from binding. Predict the downstream effects on transcription, translation, and protein synthesis. I'll see you in the next interactive tutoring session. Let's microbiology it.